Thank you for joining us today for our webinar entitled Lipid Nanoparticle Characterization, Leveraging HPLC-CAD for Lipid Quantification Assay. The purpose of today's webinar is to offer invaluable insights into the critical techniques and methodologies that are shaping the future of lipid-based characterization assays. Whether you're involved in research, development, or quality control, this webinar will equip you with the knowledge and tools you might need to enhance your analytical processes and to ensure success in your LNP projects. Today's webinar's key takeaways are to understand the critical components for LNP lipid quantifications, to learn what it takes to develop robust methods for HPLC-CAD analysis, to learn tips to navigate common challenges during analysis and avoid these common pitfalls, and along with our speakers to explore a real-world case study demonstrating quantification of lipid in an LNP packet. Before I present today's speakers, a quick overview of our company. Founded in 2005, Phosphorix is a CDMO specializing in particle-based drug delivery solutions. Located in Hopkinton, Massachusetts, just 26.2 miles from Boston, our state-of-the-art facility spans 30,000 square feet, with cGMP clinical trial material services expected to be online in 2025. Our technical expertise and experiences are working with lipid nanoparticles for nucleic acid delivery, polymeric nanoparticles for targeted delivery, and polymeric microspheres for sustained release delivery. We are a fee-for-service model for contract work, and our mission is to accelerate client programs toward success in the clinic. But before we begin, a bit of background on our speakers today. Our first speaker is Sahir Abu Aita. Sahir joined Phosphorix in September 2023 as Director, Analytical Services. In his role, Sahir provides vision and direction to the Analytical Services Group in support of the development, validation, and implementation of analytical and process control methodologies to support the future establishment of our cGMP manufacturing capabilities. Sahir brings 15 years of experience with Phosphorix in both analytical method development and quality control in both a CRMO and CDMO environment. Prior to joining Phosphorix, Sahir held the position of Quality Control Lab Manager for New England sites at Vera Nova and Johnson Matthey. And our second speaker today is Sharon Seton, Associate Director for Analytical Services. With over 20 years of experience in drug research and development, Sharon has worked in biotech, pharmaceuticals, CRO, CDMO, and academia. She specializes in developing and validating methods for small molecules and biologics, including peptides, proteins, antibodies, and nucleic acids, using techniques such as HPLC and fluorescent assays. Sharon has expertise in analytical procedure for drug delivery systems, including nanoparticle and lipid nanoparticles. She has held roles at BMS and Spectrix Analytical Services and earned her Bachelor of Science from Fudan University in Shanghai and a PhD in molecular pharmacology from the University of René Descartes from Paris. And now I turn the presentation over to Sahir, who will get our webinar started. Sahir?
Thank you, Kim, and hello, everyone. For years, mRNA has been investigated as a viable candidate for vaccine against infectious diseases such as influenza. The recent pandemic has significantly accelerated this research, resulting in the emergency authorization of COVID-19 vaccine in 2020. This pivotal moment underscored the pressing need for innovative analytical method to accurately characterize these novel products and ensure their safety and efficacy. At Phosphorix, we are committed to leading this innovation by developing a comprehensive array of analytical methods focused on the quality assessment of lipid nanoparticles and nucleic acid delivery systems. In response to the growing demand of effective characterization, we have invested in a variety of advanced analytical instruments to thoroughly evaluate the quality attributes of such products. In this webinar, we will highlight one of these methods, the lipid quantification using the charged aerosol detector, or CAD. Before we move into the details of the test method, I would like to give a quick introduction and some background on lipid nanoparticles and why lipid quantification method is important to assess the characterization of the particle. Lipid nanoparticles are small spherical structure made from lipids that act as a vehicle to deliver nucleic acid, such as mRNA, into a cell. Typically, there are four main components that makes the LNP body: an ionizable lipid, pegylated lipid, phospholipids, and sterile lipids. The ionizable lipid is a lipid that is responsible for making the electrostatic connection with the receiving cell membrane within the body. The pegylated lipid is the lipid that helps in improving the stability of the particle. The phospholipid is the lipid that helps in improving the encapsulation efficiency, and the sterile lipid, usually cholesterol, helps in improving the delivery as well as the stability of the particle. Given that each lipid has a distinct function within the LNP system, it is essential to track composition of these lipids throughout the manufacturing process and the entire product life cycle to guarantee the safety and efficacy of the final product. With this in mind, I would like to hand it over to my colleague Sharon, who will guide us through the details of this method and go over some real-life case studies.
Thanks, Kim and Sahir, for the introduction. As Sahir mentioned, the COVID-19 pandemic and the resulting mRNA vaccines have significantly increased the interest in the potential for lipid-based nanoparticles, which can carry different types of therapeutic payloads, such as RNA and DNA. LNPs are usually a multi-component mixture of lipids. Due to the differences in charge and relatively UV insensitivity, simultaneous detection of lipid packet is a challenge. Here we present a reversed phase HPLC using charged aerosol detection method. The purpose of this work was to establish a suitable rapid method for lipid profiling and quantification of the respective lipid components, as well as establishing linearity, repeatability, specificity, and working range. Several technical challenges should be considered in the development of HPLC-CAD method. I will discuss the four factors and how each of them influence the chromatography of the lipid packet. Number one, broad peak with pegylated lipid. Method development for PEG lipid is particularly challenging, which associated with its inherited property and the smallest percentage in a typical lipid packet. Therefore, choosing CAD detection method and the prop column selection is critical for addressing this challenge. Number two, carryover. In the early stage of method development, we observed that the residue of lipid peak was carried over in the following blank injection. To manage this problem, different washing solutions were tested, a combination of the strong solvents selected, and increasing the amount of organic phase at the end of the run to ensure everything is removed. Double blank injections in between the samples were also beneficial. Number three, consistency of chromatography profile. With the complexity of the multi-component lipid packet, the selection of column and mobile phase composition and choice of gradient program, etc., are factors that contribute to a satisfactory and a stable chromatography profile. Other factors to keep in mind: the age of the mobile phase and allowing enough time for column equilibration. Number four, sample preparation. How we prepare the lipid nanoparticle sample and the solvent used in the sample preparation might have an impact to lipid packet profiling, quantification, and solution stability. We need to ensure the proper grade solvent or reagent is used for sample preparation. Mindful of the previous mentioned technical challenges, we developed a reverse phase HPLC method using a modified C18 column and a combination of gradient mobile phases. The gradient can be modified to fit for specific lipid packet if needed. The CAD setup parameters are summarized in the following table. On the bottom of this slide, a representative chromatogram was obtained using the developed HPLC-CAD method. It consists of 4 peaks corresponding 4 lipids in a lipid packet. The PEG lipid was first eluted, followed by cholesterol ionizable lipid and the HELP lipid. It shows clear peaks and baseline separation of the lipid and exhibit good lipid composition profiling. A series of stage prop validation studies, including linearity, system suitability, robustness, repeatability, and spike recovery was conducted. The short-term storage stability for sample solution was performed as well. We also initiate the Foster degradation study in various stress conditions. In the following a few slides, I will talk about the details for each of the parameters. Our data demonstrated that this developed method is fit for the intended purpose. The linearity was determined over a 7-point calibration ranging from 0.6 to 40 microgram per milliliter for every lipid tested. All lipid samples were gently vortexed until each solution appeared clear. Some of lipids were heated to 50 degrees Celsius to be fully dissolved. In the up panel, the overlaid chromatogram shows 7-point calibration are superposed for each lipid. The peak areas are proportionally increased as the concentration level up, while the retention time remains unchanged. In the lower panel, the calibration curves show a linear fit across all 4 lipids with a greater correlation factor that is greater or equal to 0.995, indicating the linearity was established. System suitability was performed by injecting 6 replicates of lipid standard solution to determine the accuracy and precision of the system. The data summarized in the table indicated that the relative standard deviation is less than 1% across all 4 lipids, which meets the acceptance criteria. The repeatability of the method was assessed at low, medium, and high concentration levels, 3 replicants at each testing level. Each histogram represents the average of 3 replicants in high concentration, color in blue, medium concentration in orange, and low concentration in gray color. The arrow bar on the top of the histogram is the standard deviation. The relative of standard deviation is less than 2% across all 4 lipids. As defined by the USP, robustness measures the capacity of an analytical method to remain unaffected by small but deliberate variations in method parameters. In this study, we select the column temperature adjusting by plus minus 5 degrees C as a modified parameter. The chromatograph obtained with plus minus 5 degrees Celsius change in comparison to the chromatograph obtained using the target parameters. All three display the same profile. Variation in peak areas is below 5%, which is within the limits that produce acceptable chromatography. It indicates the analytical method is robust and reliable during normal operation. The spike recovery study was performed by adding a known amount of a single lipid to the lipid packet matrix. The actual response of the spiked sample is compared to the theoretical value. The ratio of the two sets of data is the calculated recovery. We assess the recovery at 3 levels, spiked with low, medium, and high concentrations for each lipid. The study results are summarized in the histogram figures on the right. Each of them represents the 3-level recovery for an analyzed lipid. The percentage of recovery ranging from 95% to 105% across all lipids analyzed. These results further demonstrate that the analytical method we developed is accurate and efficient. Ensuring proper lipid packet storage is crucial for maintaining the integrity of analytes. We then evaluated the different sample solution storage conditions at room temperature, 4 degrees Celsius, minus 20 degrees C, and minus 80 degrees C. The duration of the study was up to 9 days. Samples were collected on day 1, 2, 5, 6, 8, and day 9. The preliminary data indicated that 4 degrees C was the preferred storage condition among the 4 temperatures evaluated in this study across all 4 lipid solutions. We then successfully developed an HPLC-CAD method and conducted stage prop validations as shown for the previous slides. In the following slides, I will present how the method can be optimized to simultaneously quantify different lipid packets associated with different ionizable lipids. In this slide, a representative chromatogram of the 4-component lipid packet was generated using the previously discussed parameters. The chromatogram exhibits the desirable profile by its consistent retention times, symmetric peaks, peak efficiency, and good resolution among the 4 lipid peaks. When the same method applied to another 4 components of lipid packet with ionizable lipid 2, it only shows 3 peaks in the up panel of chromatogram. Obviously, both cholesterol and ionizable lipid were co-eluted. To address this issue, we then modified the gradient. The separation of cholesterol and ionizable lipid achieved as shown in the lower panel of chromatogram. Here is another sample of lipid packet in LNP formulation that we analyzed. It shows that the ionizable lipid 3 was also co-eluted with cholesterol using the original developed method. Further optimization of the method was granted in achieving the separation in this case. In a similar situation, another peak co-erosion case was observed in analyzing lipid packet associated with ionizable lipid 4. In this case, as shown in the up chromatogram, both peaks were partially co-eluted. In other words, the baseline resolution criteria was not met. Thus, the accuracy of quantitation would be impacted. Once again, the mobile phase composition was modified. Therefore, the separation was obtained as indicated in the lower panel of the chromatogram. Our next goal is to apply this method to assess the lipid composition during LNP formulation key steps to gain a better understanding of LNP formulations. LNP formulation development usually includes two typical downstream process pathways. One is dialysis, and the other one is TFF process. In this study, we compared the initial lipid packet stock solution used in the LNP formulation to the in-process samples in each key step, which are highlighted in the red boxes, along with the final LNP products. Case study 1 assessed all samples collected from dialysis process in comparison to the lipid packet solution. The first set of histogram bars represented the initial lipid composition before introducing in the formulation, so-called lipid standards. Next to it is the set of LNP1, which represents the samples collected from post-dialysis step. LNP2 is the data from post-centrifugation, and LNP3 is the data generated from post-filtration samples. Lipid composition for LNP1, 2, and 3 were analyzed and compared to the lipid standards. The calculated lipid molar ratios for all LNP samples are in good agreement with the initial targeted lipid molar ratios, indicating a successful process. This statement was further supported by the other characterization parameters, such as particle size, PDI, and encapsulation efficiency. Case study 2 is the assessment of samples collected from various stages of TFF process. By comparing to the lipid packet, there are no significant changes in lipid composition across all stages in TFF process for the analyzed LNP formulation. Their lipid molar ratios reassembled that of the lipid packet stock solution. These results are in alignment with the other characterization data, such as particle size, polydispersity, and encapsulation efficiency. All data proves that the formulation process was successful. Unlike the previous case, this case study illustrates deviation in the in-process samples when compared to the initial lipid packet. The change of lipid composition profiling intensifies in post-concentration sample, circled in red. Even after filtration, the profile change cannot be totally restored. The lipid composition deviation reviewed by HPLC-CAD method was also in correlation with enlarged particle size, elevated PDI, and decreasing in encapsulation efficiency, indicating the process performance in this LNP formulation required further optimization. To summarize, During the presentation, we demonstrated that this HPLC-CAD method is reliable, robust, and proven to be linear over the range of 0.6 to 40 microgram per milliliter. The sample solution is stable up to 9 days when stored at 4 degrees C. During the method development, we successfully addressed the key technical challenges, such as broad, pegylated peak, carryover, consistency of chromatography, and the sample preparation. This method is also proven to be easily adapted to a wide variety of lipid packets upon minor optimization. Furthermore, it allows a broad range of applications in quality assessment of various formulation processes, as well as final LNP products.
Thank you, Sahir and Sharon, for a great presentation. At this time, we'll go ahead and get into the Q&A discussion. If you could both get back on video and audio, that would be great. Great. I have some questions that came in. For the audience, you still have time to submit any questions. Just pop them into the chat box and we would get to you as soon as we can. My first question, lots of research papers are currently using ELSD for lipid analysis. Why did you choose CAD detection, and which detector is the better option in lipid quantitation?
Well, it's a very good question, Kim. I can take this one. So ELSD detector has been used in the biotech pharmaceutical lab for quite many years. However, the CAD detector is considered a more recent technology. In our lab, the reason that we choose the CAD detector is because of several reasons due to the CAD detector property. One is the high sensitivity, and the other one is the wider dynamic range, and also it's more uniform the response of the CAD detector versus the ELSD detector. So all those properties make the CAD detector a better option for accurate quantitation of the lipid during the lipid analysis. So both are the reason that we used. And then the other lab probably, because as I mentioned, the ELSD technology has been longer time than the CAD detector. So it might be used by the other research lab, but in our case, we feel the CAD detector is a better option for the lipid quantitation.
Thank you, Sharon. Our next question is this method a stability indicating method? And to what level have you forced degraded the sample?
Maybe I'll take that question, Sharon. Maybe I'll start with the second half of the question. So we did just preliminary evaluation on the force degradation at this point. We didn't carry the force degradation study all the way through yet. With that being said, I wouldn't necessarily say at this point it's stability indicating. We're using this method as a quantification method for in-process and some final product analysis with the intention of completing that study and perhaps doing any necessary optimization on the method to allow for the separation if there's any peaks that would collude during analysis. So that work is still in progress, and we'll be continuing that work to ensure that the method is stability indicating and be used for that purpose later.
Thank you. The next question we have is, can you provide details about LC condition in your lipid quantitation method?
Well, without getting too much details about the the lipid composition or the condition of this method. And what I can share is we use the column, which is the modified C18 column, and a combination of organic solvent, and with the buffer modified aqueous solution, a combination of these as a mobile phase. And we do use the gradient method versus the, well, actually, it's the gradient method that we used as a gradient program. So yes, that's my answer for this question.
Thank you, Sharon. Next question. It looks like Sahir and perhaps Sharon could both take this question. For the sample preparation, does the method require removal of payload of LNP?
Well, I can take this question. We don't typically remove the payload, but actually we see a big peak up front. But regardless, that's not interfere with our lipid quantitation. The peak will be come off like a much later. So that's not affecting. So we just don't bother to remove the payload in our method.
Thank you, Sharon. Next question would be, what is the basis of choosing 4 Celsius as preferred storage over -20 or -80 Celsius?
Yeah, maybe I'll take this one, Sharon. So based on the assessment that we've done and we demonstrated in this webinar, we did perform that evaluation over all 3 temperatures, and on top of it, the room temperature as well. So looking at the variability that we've seen and the change over time between all different temperature, the 4C seemed to be the least condition that actually the least variability that we've seen in that condition. Therefore, the choice was to go with that condition as the preferred condition. Looking at the data, if you notice the presentation on that slide, there's great variability with the rest of the condition. Now, the variability could be, sample to sample, but it seems like there's more into it, which indicates some change in the composition of the sample as the time passes through.
Okay, thank you, Sahir. Next question. What mobile phases would you suggest?
I know Sharon touched a little bit onto that. I mean, what I can say on that end is a composition of a aqueous, modified aqueous, and organic, where the gradient would you would go through the gradient program to allow for the proper separation. That's what we've seen that worked. Just to give it a little bit more of a color, maybe for the organic, there were 2 types of organic that were used as the organic phase. But it's, as I said, it's a combination of modified aqueous and those 2 organics to achieve the separation based on the sample that we're testing.
Great, thank you. Next question. How do you explain the marked loss of ionizable lipid post-concentration in the example? And did it degrade or was it lost for other reasons?
Maybe I'll take a swing at that, Sharon, and feel free if you want to add to it. I think it's I don't think it's a loss, maybe aggregation, maybe. Again, we didn't dive too deep into analyzing the reason why it might be aggregation. But for that, again, we probably would need to do further assessment to understand it a little bit better. And the reason why we're potentially seeing aggregation is the next step, which is filtration. It seems like the composition went back up indicating that the aggregated particles were filtered out. Therefore, the constant or the distribution went back to what we would expect as a normal distribution. But again, this is just speculating based on our experience, based on what we've seen from the sample, not coming from an analysis of that particular scenario.
Thank you, Sahir. Another question here is, for the spike recovery assessment, what is the sample matrix being used? And would that spiked sample go through the sample prep procedure?
I can take that question.
Sure.
Actually, this is a very good question. The spike recovery, the matrix that we used is actually the lipid packet. And the spike sample, actually we used at various concentration of the single lipid, and then we spike this into the lipid packet. We call the lipid matrix. So that's how we prepare the sample and then how we conducting the assay. So I hope I answered the question for that one.
Sahir, do you want to add anything to that?
No, I think Sharon summed it up correctly. It was an addition of a standard to the sample itself and doing the difference thereafter. Yep.
Great, thank you. We have one more. Let's see here. For post-concentration sample in CAD, would you not expect to see the peaks to be larger than dilute?
Let me think of that. For our post-concentrations, do not expect the peaks to be larger than dilute. I'm not sure if I'm getting the question correct, but that's not what we've experienced. Again, I'm not sure if I'm getting that question correctly. I'm not sure what the person means by dilute, but we did not observe any enlarge of any peaks throughout the process. Yeah. And if the person want to elaborate a little bit further to clarify it, yeah, please do so.
Yeah, we could definitely follow up on that question. I have another question here. In your presentation, the calibration curves are linear to all lipids, while lots of works in the literature indicate a non-linear curve. Can you comment on to why yours is different?
I can take this question. It's a very good question. The reason that I think a main reason for we get the linear calibration curve is because of we use the relatively lower range of the calibration concentration, which is like in the presentation you would see, it's a 0.6 to 40 microgram per milliliter. So in this range, we do observe the linear regression that we have the R-square number, which is greater than 0.995. And we would think maybe in the higher concentration of this, or we have the large magnitude of cover the more large range of the calibration standard or the higher concentration, we might see this linear regression might be not a best fit. So in that case, we might consider the logistical regression, which is probably such as the polynomial fit. But we do, in our calibration range, we do see it's a linear regression, which is correlated very well. And also we think the linear range, it's always better for the quantitation. So we stays in this concentration lower range. And the secondary, the reason that we observe the linear regression is because of we use the CAD detector. I think it's a very important factor. The CAD, as I mentioned earlier, it's more uniform, the response, and independent of the structure of the analytes. So usually the most of time, the CAD response is linear. So that's the reason that we do observe the linear regression. And then the other lab or the other research paper, they might have like the large or the high level of the concentration, which is off the linear range maybe. And also they use some of those I know they use the ELSD detector. The ELS detector is slightly different than the CAD detector in terms of the linear response. So for these two main reasons that we do generate the calibration curve in linear. So I hope that answered the question.
Thank you, Sharon. So the challenge has been accepted by Sahir, and it looks like he does want to go back and revisit that last question. For post-concentration sample in CAD, would you not expect to see peaks to be larger than dilute?
Yeah, sorry, I misread the question, or I understood the question earlier. The answer is yes, that is correct. Yes, we would expect it to be higher or larger. Now, just to clarify on the presentation, what we were looking at are ratios between the lipids. So not necessarily for the purpose of this presentation, not necessarily the actual true concentration of these lipids. So we're looking just as ratios within the lipid packet. So the expectation is if the concentration is higher, all of the lipids would go up, would increase in the same ratio relative to each other. Therefore, the composition would remain the same. Now, typically what we do is we try to prep the sample at a consistent concentration. In other words, if the sample is too concentrated, we'll use less of it to maintain the same unified, as much as possible, sample concentration throughout. So we have a better representation. But short answer to the question, that is correct, yes.
Thank you, Sahir.
Yeah.
I have another question here. How did you screen for the diluent, and what did you observe with other diluents that were not selected as the diluent of choice?
Sure. Maybe I'll take this one too. Actually, this is a pretty good question because typically the diluent is the silent part of a method development. Not a lot of attention goes to it, but in this case, I would think it's critical because we're trying to achieve multiple things here with the diluent selection. One is to ensure that we're breaking down the lipid particle, so we have a proper analysis of the lipid. Two, that we're preserving the quality of the sample so it represents the bulk material. Therefore, our screen went through multiple type of diluents, ranging from all aqueous to all organic and a mix in between, where we were evaluating what I stated before. Is it able to break the sample? And is it able to maintain the quality of the sample that represents the actual bulk material? So with that being said, we went through multiple to get to it. And, where we've been successful is a combination between an aqueous and an organic diluent.
Thank you for that. One more question here. Any suggestions for sample prep of LNP before LC analysis, and how to break the LNP?
Okay, it's a very good question. I can take that one. Suggestion for the sample prep of LNP before loaded into LC analysis, we can do the different way. One way that doing this is we use the Triton solution as we do for the ribogreen or the pickle green assay. We can break with the Triton to lysine and deassociate of the lipid and the cargo. So that's one way, but it has to be very careful. Once you use the Triton in your LC sample, that after that injection,
I would suggest that you wash several times with the organic solvent. You make sure the Triton get away from your system, and you don't want to disaccumulate, because we noticed that sometimes after several injection without washing, then your peak is getting more broad and the peak gets smaller. So that's the lesson learned away from or using the Triton in the solution for the sample prep. So yes, you can use the Triton to break the LNP, but you do need to wash several times until that disappears. I mean, until, yes, we think if you can remove totally. That's my suggestion.
Thank you, Sharon. Please, audience, if you have any other questions, we do have a few minutes here. Please pop them into the chat box. But one more question here is, have you observed any impurities during the sample solution stability study? And what is the observed variability within the method?
Maybe I'll take this, Sharon, but again, feel free to jump in and add on. As I indicated through this discussion here, we didn't dive too much into the impurity profile. We have not performed the full force degradation yet. So relying on my memory here a little bit in terms of the impurity observed, we did observe some impurities that were resolved from the main peak as we were going through the solution stability study, but we were not quantifying it as, just because we were not, the method is not looking at the impurity at this point. It's looking for just the quantity of the lipids. So yes, we have seen it, but minimal with some enough resolution, but we were not quantifying it.
Thank you, Sahir.
Yeah.
And I think we have one, let me see here. Can you provide details about LC condition in your lipid quantitation method? I think that might have already been.
To some level, again, I believe during the presentation, Sharon went through some parameters for the CAD detector parameters, where that's been set up. In terms of the rest of the conditions, it becomes more of, identifying the gradient based on your sample. So it would be very specific to the sample that is being analyzed. But as I said, the CAD detector setup itself has been presented in this presentation. The other factor maybe that I can probably share is the column temperature would need to be alleviated, again, to allow the sample to flow through and not gel through the traveling within the LC.
I can jump in, and also, like we shared earlier, the column that we used is a modified C18 column. It is a C18 column, but it's a modified C18 column. That's some additional information that I can share with.
Okay. Got. What is the column load for the analysis? That's.
I'm not sure about the question, the column load. Do you mean the injection volume, or is that what you mean?
Yeah, maybe he could clarify.
Yeah, if the person can elaborate a little bit further, but if it is, yeah, I think we shared.
I think if it's if this is refer to the column load, that the column volume is, we can share between 5 microliter to 10 microliter each injection.
Okay. Oh, he said the column load in terms of the amount of analytes.
Yeah.
Yeah. So we try to get it, as I mentioned, the calibration curve for each lipid is around it is the ranging from 0.6 to 40 microgram per mil. So in the lipid packet, each of those, because it's a component has the different level. For example, the ionizable lipid has the higher component, and the PEG lipid usually has the lowest. So we have adjusted this into, for example, if it's a lipid pack, typically we do 100x dilution, or sometimes up to 200. And it depends on which if we want to see the full panel of the lipid pack, that's how we go to, because you have to consider each of those lipid. Or with specific focus on the ionizable lipid, then we also have the high dilution. You can go to like a 80x dilution or 100x dilution. But if you want to focus on the pegylated lipid, which is a very low level in this composition, so we have to do less of the dilution volume, which is typically what we do is a 10x or 20x. I'm not sure if this answers your question, or how much it varies depending on which component you want to specifically analyze or in general.
He said, thank you for the insights. So I think he answered his question there.
All right.
We've got another question here. Are there any complementary approaches one can use to compare data from the HPLC-CAD method?
Maybe I'll time into that. So I would say yes. Again, they're all the ones that I can think of, at least at the moment, are all LC based, but definitely, like we've discussed, or maybe came up in a question, ELSD detector could be another approach. Definitely, I would think mass spec would be another approach. But again, in both of these cases, they're HPLC-based type of analysis, just using different type of detectors. Now, with lipids being, doesn't give a UV absorption, the selection of the detector is, I would say, is limited to these type of detectors.
Okay.
Yeah. Each of those has a pros and a cons, depends on which method. The mass could be a good method, but it's more complicated and it's more high maintenance a lot of times. So we choose the CAD detection, which is that works well for us, and it's I would say low maintenance maybe compared to the other method.
Correct. Yeah.
Okay, I think we have one more. See here, do you see this one?
Yes. Yep.
Okay, so this is our last question. If the formulations include additional ligands such as small molecule and antibodies, can HPLC-CAD still be used?
I would say, again, thinking of it as we're speaking, I would say yes. But to be honest, in our approach, we've been approaching it differently. We've been taking the analysis of the antibody or small molecules over to the UV. So it's a completely separate approach. So we'll do the lipid quantification on the CAD. Then we'll do a different method with a different sample treatment to analyze for any other component, just such as antibodies or small molecules. So my thought is, initially, it would say yes, but again, I would think it would be easier if we separate those two into two different methods. Again, the lipids would be on the CAD, and the rest of it would be on an LC still, but using a UV detector.
Yeah, I would say also maybe case by case, depend on what type of the molecule you're working on. Small molecule is a lot of things. So, I mean, it all depends. Then you have to test your method to see whether or not you can use the method, or you have to tweak your method to fit specific what are the analytes you want to quantitate.
Great. Great. Well, that is all the time we have for today. I did want to thank everyone for joining us today. And if there are any questions, please feel free to contact us at bd@phosphorix.com. And I appreciate your time. And within 24 hours, you will have an email sent to you with the recording of our webinar. Again, thank you very much for joining us and have a great day. Happy Halloween. Take care.
Thank you.
Bye.
Bye.